Hey everybody, we're going to jump into our first set of notes for this unit on Mendelian genetics. We're going to look at how traits are inherited and how they are expressed. We have the little information here on Mendel. He was an Austrian teacher and a monk. He actually wasn't a scientist. He worked in the mid 1800s, and he really was just curious about how things worked. He was kind of like the naturalist at the monastery, and did a lot of thinking about how the natural world worked. And tended to the gardens, and he started noticing a lot of pea plants. But hang on one second, I need to pause this real quick. Okay, so I'm back. So the perceptions of genetics worked a little bit like this, where if you look at this pedigree, and we're going to be getting into some pedigrees, right, where you see the parents, you see their offspring, and then you see the offspring's offspring, and so forth, right? This is generational. It's like a family tree, but we'll be looking at pedigrees here soon. Um, this was called the blending hypothesis, where every offspring had a blend of the traits from its parents, and that sounds pretty good, right? That we get a blend of our mom and our dad in our genetics, right? That sounds pretty good. Unfortunately, that is not right. That's not exactly how it goes, because if the blending hypothesis were true, then pretty soon everything would look like this, right? There would be no variation because things would keep getting diluted and diluted and diluted all the way down, and you would keep getting all the same thing, and that's not exactly accurate, right? So, didn't work quite like that. So, as we get into this, we need to look at model organisms. So, he used peas, and peas are good model organisms. Um, you need things that are small, that are easy to grow, easy to maintain. You can control the mating. You get lots of offspring. The traits are easily seen, easily distinguishable, and you get quick generation time. So, if you can find organisms that fit into this, you can definitely work with them in genetics, right? Because if things take a really long time uh, to to mate and to have babies and all that sort of thing, and they don't have very many, then it's going to be really hard to tell what the genetics are doing. So there's lots of model organisms. Maybe you know some others besides peas. There's basically one or two in each lineage or each type of organism. So, like for insects, and you have the fruit fly, right? Fruit flies are a very experimental organism, right? We use those. There's a worm called C. elegans, which actually the lab that I used to work on in the genome lab. I worked at the Washington University Sequencing Center for genomes for a while before I was a teacher, and we worked on C. elegans, which was a worm, and we did do the fruit fly, and we worked on zebrafish, and I'm trying to think what other ones we worked on. Mouse is another model organism for mammals, right? So there's lots of different ones. Oh, we did a weed, a plant called Arabidopsis. And there's lots of literature on all these organisms because they've been tested any which way you can think of. Okay, so that's what we use model organisms for, to test and to see how they do, and we can apply that knowledge to bigger organisms like humans. So this just gives you a little bit of background on how the pea plants work. If you didn't know, pea plants actually do have flowers during their life cycle. So you can see the male stamens there and the female stigma, and so as the flowers kind of then drop off, you can see as the pictures go around the side here, you end up with this pod at the end, and then the peas are in the pods. So you can control a little bit about what's going on, and you can actually take certain aspects of these flowers, and you can cause them to mate or cross with other flowers, right? They would take paintbrushes and like touch the pollen of one flower and then move it over to the ovule of the other flower so that they could cross particular flowers that they want. Okay, you can get cross pollination of pretty much anything you want, and sometimes self pollination. It doesn't happen a lot that flowers pollinate themselves, but it does happen, and sometimes you can force that to happen. So back here real quick, I went a little quick. The P1 generation, the F1 generation. P is just parent generation, like your original. The F stands for filial. You don't need to know that word, but it's really just the first offspring generation. And then F2s would be, under that, and then F3, F4, all the subsequent generations. So Mendel's methods, was to establish a true breeding line for a trait. And a true breeding line is a line or traits that you know what you're going to get. So for him, he would have all purple flowers and all white flowers, where if you cross purple and purple, you'd always get purple. And if you cross white with white, you'd always get white. There were no like heterozygous ones in there, right? These were true breeding homozygous dominant or homozygous recessive. So he started with those and had true breeding lines. Then he started crossing those lines, and then he just started counting, right? You just start counting the offspring and what you get. And so this one right here is just purple versus white flower color. So, here's your parent generation up here, and he took purple flowers with white flowers. Then the first generation, he got all purple flowers. He'd never get anything else because, as you might imagine, this is a big P over here, and this is a little p on this side. So you're always going to get this, and so you would always get a purple flower on that generation, right? But when you start crossing these, right, and this is self or cross pollination, so especially when you start self pollinating and you do big little cross with big little as far as the letters, now you're going to start getting white back into the mix, and that really, probably had to freak them out. That how did I cross two purples and I got a white? So they had to start working out how that went. Okay, so, and you can see they're just counting 705 purple to 224, and that ends up being about a 3-to-1 ratio, right? So here's, a lot of different things. Here's the color. Axial or terminal is where the flowers are, right? So this one, they come out the side. This one, they're at the very end. That's the terminal end. Yellow versus green pods, round versus wrinkled pods, inflated or constricted, green, yellow, tall and dwarf, like all these different things. And here's all your ratios right here. Never exactly 3-to-1 because look at how many they were testing. They were testing thousands of them, right? And so you're not going to land on a perfect 3-to-1 every time when you're testing thousands, but darn near every one of these is very close to 3-to-1. So they knew something was up, right? There had to be a pattern. There had to be a reason why. So his conclusion was there was two alleles, okay? Meaning you had to get one from mom and one from dad, right? And so they had to exist at the same place, and they didn't know all of the terms that we used back then, but there had to be factors or units that were then passed on. So he concluded there had to be a dominant allele and a recessive allele, and that one could hide the other. Okay. So then we get into a couple of laws here. Mendel's law of segregation states that alleles will separate, and each gamete will only receive one allele for each trait. Okay, this process is random, and when does this happen? So this process is random, but remember, they do separate, and it just kind of has to do with how they line up. Remember how we used to do this or do this, right? And that would be at metaphase in meiosis, right? Especially in meiosis 1. And so that happens, and that's going to guarantee that each gamete is going to get one of each allele as long as there aren't any super big issues with mutations and things. That the alleles will separate, and each gamete will receive one allele for each trait, because you're ultimately going to get, if it was a human, you're going to get 23, right? In these final 4 cells down here. Remember, way down here, that these 4 are going to be haploid, and they're just going to get one copy of every chromosome of every trait, right? There's not 2 copies. So that's segregation, just that every gamete will get one of everything. Law of independent assortment states that alleles for separate traits are passed on independently of each other, right? So as long as the traits are unlinked, and we'll get to linked and unlinked, but basically means separate chromosomes are far enough on a particular chromosome that they don't travel together. Okay, and I know that might be a little confusing so far, but not only today, but in another set of notes, we'll talk about linked a little bit further. So independent assortment really just means that if you're tall and blue eyes, that not every tall person has blue eyes, and not every person that has blue eyes is tall, because those 2 traits are independent of each other. They don't have to travel together, right? So we don't have like a set of criteria that, if you're tall, you're also, this color eyes and this color skin, and you don't have freckles and whatever, like you have all these things. Every trait, every single thing assorts independently from each other. And that's why we get all these different combinations. It's what lends itself to variation, right? So independent assortment, independent assortment is really what's going on over here, where we produce this many gametes, right? On the egg side and on the sperm side, we have this many choices from these parents right here. Okay, where over here, if we have the same thing, if they did not uh, independently assort, so down here you can see dependent assortment, okay, then that means you would only get these two and these two options. And so that would mean you only have this 3-to-1 ratio, but that's not what they were seeing. They were seeing all this variation on this side. And so they figured out that things had to be separating, right? So this is yellow round, and this is green and wrinkled over here. And so you would only have a few options, and you would only get the yellow round and the green wrinkled. Basically, you would only get what the parents were, right? But that's not how it works. We get other options over here. We have other phenotypes that are different from what the parents are. And so that lends itself to independent assortment. And so how things, each trait assorts independently. And you can be yellow and round or yellow and wrinkled, or you could be green and round or green and wrinkled. And then, many more options. So the two laws here, law of segregation and law of independent assortment, are happening during meiosis, right? And so a lot of that's going on at meiosis one and in metaphase when they're lining up, and that's going to dictate, as long as things separate out, you're going to be able to have one copy of everything in those gametes at the bottom, and that they're not all going to be the same. So recombinant offspring, I've talked to you about this before. Independent assortment can lead to combination of traits and offspring that are different of the parents. So remember what it means to be a recombinant offspring. So in this case, you have a parent that is green and wrinkled, right? So it's green and wrinkled right here, and up here you have yellow and round. So when you do the cross real quick, you can see that this is a yellow and round one, and this is a green and wrinkled one, but that's what the parents are, right? This one is the same as that parent, this one is the same as that parent. But these two over here are the recombinant ones. They have different, right? Yellow and wrinkled and green and round are not like the parents, so we call those recombinant. Okay, they are different from the parents. So this is the death of the blending hypothesis. Mendel kind of killed that hypothesis and really said that families are just another example of recombination, right? When you think about your family, there's all these different variations that are going on, and you're similar, but you're not the same, right? And you're not all, things didn't dilute out where your siblings and your cousins and everybody are kind of the same. There's still all these differences, okay? So a few genetic terms here. Be sure you know how to apply these. I've mentioned these already before. They've been going on in the Punnett square stuff we've been going on, but homozygous versus heterozygous, dominant versus recessive, gene versus allele, monohybrid versus dihybrid. Test cross we haven't really talked about yet. Let's see if I can come up with an example for test cross. So if you had two red apples, right, or just two apples, let's do tomatoes. If you had two red tomatoes, and big R is red, right? So big R is red. And what if I handed you a tomato? If I handed you a red tomato, how would you know if that tomato was this or if that tomato was this? Would you really be able to tell by just looking at it? Probably not. So how could you do a cross and figure that out? What would you want to cross it with to figure out if that tomato I gave you was big R or big R little r? How would you know? Well, if you do a test cross, then you are going to cross it with the recessive, right? Because this is a guarantee, right? If I take this one, if I do this cross, big R by little r, I will always get red. Always, 100% of the time, I'll get red, right? If I do this one, if my tomato happens to be that, and I cross it with this, now I'm going to get red part of the time, about half the time, but then I'm going to get yellow or the off-color tomato about half of the time. Okay, I'll just call it yellow for now. I'll get yellow. And so I'll know by which colors I get. If I get a mix of colors, then I'll know my first tomato was this. If I always get red, I'll know my first tomato was big R, big R. So a test cross is using the homozygous recessive option to figure out whether you have a homozygous dominant or a heterozygous option. Okay. Okay, so we're going to also work on some probability stuff here, and this is actually where I kind of stopped the slides in class, and we watch a video from Paul Anderson, the Bozeman biology guy, because he has an awesome video on this and does probably a better job than I would explaining it. So I'm just going to click through these now, but I'm going to post the video, his video, along with mine. And you need to go watch that video. It does really well at explaining two things: the rule of multiplication and the rule of addition. And this helps you deal with bigger events, okay? Bigger things that you really wouldn't want to do Punnett squares for. So for the rule of multiplication, these problems have the word and in them. Like, what are the odds that you would get, this outcome and this outcome, okay? So independent events in a sequence. So the video will help you out with that. And so here's kind of an example, but he does plenty of examples in there. But these slides are here for you as well. And then the rule of addition is an or. What's the chance that I would get this event or this outcome? Like this outcome or this outcome. Okay, what's the chance? And when I roll a dice, what would be the chance of getting a 2 or a 5, right, on a 6-sided dice? So those type of questions. What are the odds in pea plants that are heterozygous for purple color produce a heterozygous offspring? So these sorts of questions, and these are mutually exclusive events, these rule of addition ones, meaning they can't happen at the same time. Like you can't flip a coin and get heads and tails, right? It has to be heads or tails. So those are these examples are problems like that. So again, watch the Paul Anderson video and he will go through all of this with you with great examples, okay? And this is a practice problem for what we're talking about. So when you get a cross like this, a tri-hybrid or a quad hybrid or something bigger, these way to deal with the probability is way easier than dealing with a Punnett square. So make sure you watch this video. Make sure you practice how to do these because you just might see them again. Okay, so that's all I got for you for now. There's some practice questions for you. The slides will be posted, my video will be posted, and Paul Anderson's video will be posted. But you should check those out. Okay. All right. Have a good day. Talk to you later.